Okay, so control of the internal environment. What's going on in here? Hopefully you have this printed out or ready to take some notes on it or whatever. So here we go. Here we go. Mostly what we're going to be talking about is homeostasis and steady state, and a little bit about like biological control systems and negative feedback. If you already know what a biological control system is and what negative feedback are, then you should be good for this. That's really the main testable material that I'm going to have throughout the course of this lecture set. There's only like 20 some odd slides in here, so like let's go and start getting through those as fast as we possibly can. So right here, homeostasis. The typical definition is the maintenance of a constant normal internal environment. Now, all of us being humans, we never really have a fully normal constant environment. It's more of a dynamic constancy. So really, like I'll show you a picture here soon. Mean arterial pressure, it kind of oscillates up and down based upon just kind of your breathing and your second to second like level of like neurotransmitters or hormones floating through your body. So really, I think a better way to think of it is dynamic constancy. But that's all that is. Now, in regard to exercise, a much more important thing is a steady state. And this is going to start coming up a lot in labs to come. So if anybody like remembers the 3 energy systems that we were talking about in the lab yesterday, how it takes approximately 2 to 3 minutes for the aerobic or oxidative system to essentially kick on, we're effectively going to have staged or graded exercise tests here soon to where we maintain a given like physiological level that isn't homeostasis. And that is going to be termed a steady state. So here, let's read this really quick. So it's a physiological variable, is unchanging, but not necessarily normal. So it's kind of a balance between the demands placed on the body and the body's response to those demands. So say like blood pressure. Whenever we start exercising, our blood pressure goes up. But our body is trying to maintain us at homeostasis as best it can. But if we're running, say, 5 miles an hour, 6 miles an hour, whatever, we can't maintain your normal heart rate or homeostasis that you're at right now. We have to maintain a higher level. And whenever we're at that level, doing like constant continuous exercise, that's what is referred to as a steady state. Hopefully that makes sense. Graphically, this is what it essentially looks like. Here on the y-axis, it's showing like body temperature. Like we're not going to measure body temperature in class ever, so just see that as heart rate, blood pressure, respiration rate, just about anything this could be. So if an individual is like exercising, if you increase the intensity, meaning you go faster, or you increase the incline, or you increase the weight on a bike, or something of that nature, our whatever variable of interest that we have, it's going to go up and essentially meet like a ceiling to where your body is able to maintain that level. Does that make sense to everybody? Right? Hopefully so. If you took health appraisal, we did like quite a bit of this. So like that's really the main thing I want you to understand about steady state. Now this picture is kind of illustrating the dynamic constancy of homeostasis that I was talking about before. So mean arterial pressure, hopefully everyone knows this. We have like a systolic and a diastolic blood pressure, and we do some type of math to figure out what the average pressure is like within our vasculature at any given moment in time. So it's always going up and down, up and down. And really, this line that's going through it, that's more or less the like homeostatic line that is going through like the peaks and valleys of our blood pressure. So something you can actually like pay attention to, if you ever take your pulse rate, you can feel a quickening in the beat anytime you breathe in and a slowing in the beat anytime you breathe out. Has anyone ever done this? Well, why don't you do it now, right? So carotid, everyone, right? Oh man, like mine's going super fast right now. This is great. All right, so like breathe in, breathe out. Right, you feel that, DJ? Yeah? Oh. Oh, you answered. That was just like my mother. Cool. Great. Perfect. Essentially what that is, that's controlling two different aspects of your autonomic nervous system. So like whenever you breathe in, there's slightly more sympathetic or fight or flight system, and whenever you're breathing out, a little bit more parasympathetic or rest and digest type of system. So that's really what's going on virtually with every breath we take. You know, every move you make, right? The police. So, okay, moving on. I'm watching you. Okay, cool. Like here, a couple more things. So homeostasis, there's definition there. This term steady state, this is probably a slightly better way of looking at the definition of steady state. So if you don't have this, make sure that you have all of that taken down. Everyone good with this so far? All right, I'm trying to go quickly. Am I going too fast for anybody? Right? Fairly simple material in here. All right, control of systems like in and of the body, right? So we do have kind of intracellular signaling, intercellular signaling, between organ signaling, and just like really whole body signaling. This top thing right here, intracellular control systems, that might be a good listing question on the exam that's on the 24th. I don't know, like I might be aware of that. So effectively, our intracellular control systems are really concerned with these three main things. So the first one is essentially like net protein accrual or net protein synthesis. So at any given moment in time, we have like some catabolic or breakdown reactions happening and some synthetic or anabolic reactions happening. So ribosomes are always making more proteins, but like depending on like kind of the overall level of like energy, like ATP or glycogen or what have you, there might be more breakdown or more synthesis. Everyone good with this? That makes sense? So really quick, what's the anabolic like hypertrophy signaling pathway? What did I tell you all to know? Okay, so like someone give me the first three-letter acronym. Close. PKB or AKT. Either one is good. Yeah, so, oh, no. Like, hey, if you want to be British, Wade, that's fine. That's cool. Like, PKB or AKT, what's after that? mTOR. Yes, Barrett. mTOR. Little m, big TOR. What's after that? P7? 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 P70 S6K, right? P70, little p, big 70. I don't know if you can capitalize numbers, but we will in this class. S6K. S6K. And the S is capitalized, and I think the K is as well. So, and then that leads to protein synthesis. So that's at least one side of that. And if anybody remembers like AKT or PKB, if you want to be Wade and or British, then that actually inhibits protein breakdown from taking the FOXO from the nucleus. So hopefully people recall that a little bit. So that's one of the aspects of the intracellular control systems is really just the net protein synthesis that's going on. Another thing is energy production. So in bioenergetics, we're going to get into this like very in depth, or at least somewhat in depth. That's effectively just the production of ATP, the maintenance of that. And the next thing, maintenance of stored nutrients. So whenever we think about stored nutrients, we really mostly only store carbohydrate in the form of glycogen and fat. So in the form of like intramuscular triglycerides. Like we'll talk about that whenever we get there, but that's really neither here nor there yet. Organ systems, pulmonary and circulatory systems. Really, what I want you to know about that, kind of with the integrative like biology of exercise, they're almost always like the other energy systems, so the pulmonary system or the cardiovascular system, are essentially responding to what's going on in a muscle. So if we are producing a lot of CO2 in the muscle, we have to deal with it in some way through the cardiovascular and pulmonary system. Everyone good with this so far, right? Now, like what I'm saying specifically, that's not going to be on test, but I think it is important. Moving on to right here. This, I would really know this. This is very easily testable material. So biological control system. This is effectively a negative feedback loop, but there's three aspects of it that I want you to know. So here, I'm just going to read this to you. It's so a biological control system is a series of interconnected components that maintain a physical or chemical parameter at a near constant value. So near constant value, we think of that as either homeostasis, you know, whenever like doing the whole rest digest thing, or at a particular steady state whenever we're exercising. Everyone good with that so far? So like keep all of that integrated in your mind. The components of it are sensor or receptor. You can call it either one that you want. I'm fine with that. I think I lean a little bit more towards sensor. And then after that, control center. And then after that, effector or effector organ, as it will frequently be called in numerous textbooks. I don't believe our textbook calls it that, though. Okay. So here, there's going to be a pictorial representation of how all of this works here on the next slide. So this, I would essentially be able to understand what's going on here. And this is almost every system within the body. So there's frequently a stimulus. In our case, it's mostly going to be exercise that we're interested in. Mostly going to be exercise. So like a decrease in pH, increase in CO2, decrease in O2, increase in heart rate, increase in calcium ion concentration within a muscle, whatever we're thinking about. Whatever we're thinking about. There's something that is going to sense a change in that internal environment. Typically in the blood is what we're going to be thinking about there, but also within a given muscle cell. Then after that, there's going to be a control center, which is essentially going to assess the threat, and then respond to the stimulus through some type of effector organ, or effector cascade, or something like that. But overall, that's what I want you to understand. The stimulus sensor to control center to effector, and then a return to either homeostasis or a balancing of some type of steady state. Is everyone good with this? Right? If you haven't had me before, effectively, memorize this. That's what I'm saying here. So negative feedback loops. So most systems in the body work through a negative feedback loop. So here, that's the last point right there. So most control systems work this way. So it overall, the response reverses the initial disturbance and brings you back to homeostasis or brings you closer to homeostasis. That's more or less what that is. So like here's a bit of an example here. So carbon dioxide. So there's an increase in extracellular carbon dioxide. So overall, like we make that at the very end of like, I don't know, glycolysis or like in the mitochondria. We make more carbon dioxide. That triggers a particular receptor that sends information back to the respiratory control center. You know, frequently somewhere around the hypothalamus. I'm not going to like test you on any of this stuff, but that's all right. Then respiratory muscles are activated to increase breathing. So we begin like hyperventilating or breathing harder to get CO2 out of your body to bring us back down to a more normal level. So overall, that's kind of just what happens, right? Hopefully this makes sense. Like everyone who's ever like started exercising, then you started breathing harder. There you go. It's a really fancy way to talk about it. Okay, positive feedback loops. There really aren't many of them, and I'm not going to assess you on any of them. But if you ever get into like, I don't know, any like life cycle physiology, the common example is always childbirth. So just, I don't know, be aware of that for other classes. So, right? Okay, cool. Which is kind of weird because like, isn't this kind of a negative feedback loop too? Like they start growing the baby and then like they try to get it out of them and then they come back to normal physiology. So I still see this as a negative feedback loop, but I'm sure that like some life cycle people will fight me on it. But you know, unless you're 6 feet tall and 340, good luck. Okay, next thing. Gain of control systems. There is one particular equation that I want you to know with this. And we might do a little bit of math on the exam. Now, you should not need a calculator for this. Just, I want you to be aware of how this works. So, gain of control systems. So this is the degree to which a control system maintains homeostasis, or essentially its aptitude to maintain homeostasis. So systems with large gain, so a large number, is more capable of maintaining homeostasis than systems with low gain. So there's some systems in the body that are extremely tightly regulated. Like one thing, for example, is the amount of calcium circulating through your blood at any given time. So if the level of calcium in your blood changes too much, it's a particular like ionic charge. So like your heart could stop if it gets like too out of whack. That's normally the example of like a tightly regulated system. Other things that have like a large gain are your cardiovascular system or your respiratory system. Like you can go from like essentially not breathing for a whole minute to breathing a bunch if you're sprinting, right? Your heart rate can go from like, I'm sure some of you have heart rates like in the like upper 30s maybe, like more endurance type of individuals, like I don't know, Jordan maybe. And that can frequently go up like 200 or even a little bit higher for some individuals. So there's some systems that are tightly controlled and other systems that are not. So here is the equation that I want you to know. So gain is equal to correction divided by error. So we're going to use an example of mean arterial pressure in this case. So just think about like how much pressure is in your arteries and veins and stuff, right? So typically, a mean arterial pressure is normally around like 90 or so, but just to make math crazy easy, and I'm going to try to make it crazy easy on the exam for you, let's just say it's around 100. Mean arterial pressure is 100 like millimeters of mercury, or whatever unit that is. Oh, it says mg, milligrams of mercury. That's funny. Okay, yeah, all right. So that's hg. My bad. Yeah. You see, type slower, everybody. Right? That's as you can see, I get excited. So MAP, not mg, mmHg. Let's say it's there. A stimulus, say deadlifting, could get you up to 175 mm. See, I corrected it right there. I don't know how that happened. It could get you up to that. But the body controls it so that your mean arterial pressure only gets you up to 125. Now, here's a question that I'm sure that you didn't even have in your head, but maybe you did. How would we know that it would get up to 175? Right? Oh, bingo, you were thinking about that? Okay, cool. So do you know what a baroreceptor is? Okay, so a baroreceptor is a receptor that essentially senses like tension or pressure, so barometric pressure, right? So baroreceptor, that's how that works. We can put in like a baroreceptor like antagonist or silencer, like essentially a drug, to then make it to where it doesn't control it. And then it would shoot up really high. But like whenever we take out the baroreceptor like antagonist, then it maintains just 125. Everyone good with this? Right? Now, do you need to know any of that for a test? Of course not. Well, not yet, at least. So the correction is minus 50. Does everyone see that? So 175 minus 125, that's negative 50. Is everyone good with that? Right? Subtraction. Good. Okay, moving on. Now, here, one thing I'm actually less convinced on is so I read 2 chapters in 2 different textbooks like last week sometime. I'm not actually convinced this is negative anymore. Don't worry about the positive or negative. Just be good with the correction and error. All right? So how this works, the correction aspect of it, 125 or 175 minus 125, right? So that effectively is the correction of 50, but the error is 25 because homeostasis was 100. Does everyone see that? And now we're maintaining 125. So that's effectively how that works. Does everyone feel comfortable like you could do this math? Right? Good? On the exam, it might look very close to this. Cool? Good? All right. If you can't figure it out, come talk to me like either in office hours or lab or something like that. All right, good. Moving on just a little bit. Just a couple of examples of of homeostatic control. So the regulation of body temperature is a fairly common one. So thermoreceptors send a message to the brain. Response is typically the skin, blood vessels like dilate, and then we start sweating, and that overall regulates our heat, right? So the whole like evaporative, like heat loss, that kind of maintains our body temperature at the typical 98.6 or the 37C or whatever it is. Regulation of blood glucose is another relatively large one used in the endocrine system, particularly the beta cells of the pancreas. So it requires the hormone insulin. So after you consume food, like we normally have a given level of blood glucose, so around like 85, 90 milligrams per deciliter, whenever you're at rest, haven't eaten anything, fasted for a little bit. And then it can shoot up to hopefully below 200 milligrams per deciliter, because if not, then we might diagnose you as a diabetic. Then the pancreas senses that essentially and releases insulin to open the door into the muscles and the fat to get the glucose into those areas so it gets out of your bloodstream. So that's kind of an example of a negative feedback loop. All right. Here, essentially just a picture of how all of this goes one way or the other way. I think everyone understands the overall effect, the like sensory control center and like effector organ getting us back to homeostasis. So I'm not going to talk about this too in depth. Like either read in your book or read on this slide to get more of an understanding of this, if this like piques your interest at all. One thing here, the failure of a biological control system, two things that, well, I guess one thing that I want you to know about it. Whenever it fails, it typically leads to either disease, namely chronic diseases like diabetes, for example, disease or death. So write that down. A failure of biological control system leads to disease or death. Everyone good with this so far? All right. And essentially the classic example here, in type 2 diabetes, the effector organ is the pancreas, the beta cells of the pancreas in particular. So it's essentially not able to send out insulin to bring blood glucose back down to homeostatic levels. So whenever people with type 1 diabetes consume carbohydrates, their blood glucose goes up really high. There's all sorts of sensations in like the blood-brain barrier, the hypothalamus, everywhere that you could think of a sensation to notice this. And the signal going towards the beta cells of the pancreas, the effector organ, it can't send out the insulin to have the desired effect of lowering blood glucose. Everyone good with this? Right? I know I'm talking fast, but like you can watch the YouTube video and slow it down, right? Has anyone ever done that? I bet my voice sounds stupid, really slow. Okay, awesome. Moving on from there. A couple of things. Not really too much here that I find abundantly testable. I have asked you this question before, like exercise disrupts homeostasis in all of these different levels. Here, really quick, let's talk about these again, just seeing like what you know, right? Whenever we start exercising, what happens to pH? Up or down? So hydrogen ion concentration goes up, right? So pH being like the negative log of like the hydrogen ion concentration, pH goes down. Yeah, so, oh no, you're good, you're good, you're good. I completely understand. So pH goes down, O2 goes down, CO2 goes up, and temperature goes up. Unless you're exercising out in this environment, then good luck. So there, read the slide if you're more interested in a couple of these things. Like I'm not going to quite talk about environmental adaptations to exercise yet. Hopefully we will get there at the end of the semester. But these first two words are frequently used interchangeably, and I don't want you to ever get them wrong. So adaptation and acclamation. Here, let's talk about acclamation really quick. This is essentially adapting or being acclimatized or changing your ability to deal with the environment. So, like if you start exercising in a hot environment after doing that for a fairly long period of time, your sweat turns out to be just water, has less like salt in it, so you hold on to salt a little bit better. You tend to start sweating a little bit earlier to maintain your body temperature. You don't need to know any of that yet, but that's kind of what acclimatization is. So I want you to think about acclimatization or acclamation as adapting to an environment, typically an outside external environment. Adaptation is very related to all the genetic stuff that we've been talking about. So a change in structure or function of a cell or organ system, and it results in an improved ability to maintain homeostasis. Let's think about the endurance signaling pathway really quick. So not PKB, but what did you say earlier, Wade? AMPK, right? What triggers AMPK is a lack of ATP, or energy within a cell, right? So AMPK getting triggered, that's going to go trigger what's next? Starts with a P and ends with a GC1 alpha. Okay? Cool? I'd know that, like super well. Okay, PGC1 alpha. Then that's going to act as a transcription factor, go to like the nucleus to start making more like mitochondrial stuff. So the mitochondria essentially is the big thing. It's the powerhouse of the cell, right? Everyone knows this, right? That is what makes ATP the best inside of the cell. So adaptation, here's how I want you to think about this. If we have like a lack of ATP within a cell, so our cell adapts making the stuff that is better at making ATP, right? Like that's essentially what endurance exercise adaptation is, right? Like whenever you're like running, you need more ATP to like run better, right? Like everyone understands this, right? So then you don't have a lot of ATP, and then you can make more of it. Okay, moving on. Cell signaling. This is effectively just the communication between different cells or cell types. Here's what I want you to know. If you know Latin or Greek, all of this will make sense. So intra, that means within a given cell. So a chemical messenger inside of a cell triggers the response. So how I was talking about AMPK, PGC1 alpha, mitochondrial biogenesis. That's an example of an intracrine signaling cell cascade. Everyone good with this? Because it's a detection within the cell, and then the subsequent signals, you know, like within this whole biological control system, are all occurring within that cell. Next thing, juxtacrine. So has anyone ever heard the word, like it's frequently used in English classes, juxtapose these two different opinions, like whenever you're writing your paper? You know, compare these two, or like they're next to each other or something like that? I don't know, I never did well in English. So that's a chemical messenger passed between two connected cells. So juxta essentially means next to. Frequently this happens in like the whole like cardiovascular system, like endothelial cells. Like if one endothelial cell like dilates, then the one next to it will get a signal, like a nitric oxide signal, to then dilate. Because, well, like if we need more blood flow in a particular direction, that like essentially capillary is going to open up, and then the like next endothelial part of the capillary will open up next. Autocrine. This is a chemical messenger that acts on the same cell. And I'll talk about that like here on the next slide. I'll talk about autocrine signaling, because there's really only one good example that I have of it. Or at least that is like makes a lot of sense to me. Paracrine signaling, chemical messengers act on nearby cells. So say you're contracting a muscle. We actually secrete different things called myokines, or just cell signals, to like nearby cell types to open up blood vessels to get more fuel to that particular muscle that's contracting. So that's an example of paracrine signaling. Endocrine signaling, I think everyone understands the endocrine system at least somewhat. Like hormones circulating through your body, right? I think that's an important thing. Now here, if everything I said didn't make sense, these pictures should make sense. And actually, prior to class, Mindy asked an awesome question. On my exams, like it's mostly not multiple choice and true false type of stuff. It's mostly short answer. I like pictures. I like pictures a lot. So if you're not good with words, like how I'm sure all of you can tell I'm not good with words, draw pictures, and I'm cool with that. If I can make sense of the pictures you draw on the tests, then that's perfect for me. Cool? Everyone good with that? All right. So back to here, the autocrine, the C right here. Probably the biggest example of that, though we've talked about in class already. Does everyone remember myostatin? Like that whole like frame shift, and then you have the really like jacked up like dog or the Belgian blue cow. Myostatin in a muscle, whenever the muscle starts getting like too big, as if that were a thing, right? I thought that was funny. I planned saying that. Okay. All right. Myostatin will be secreted by a given muscle cell, go out into the bloodstream momentarily, and then bind to a receptor on top of it to stop muscle growth. So that's an example of autocrine signaling. Okay, cool. Moving on here. This next slide is in relation to acclamation. And there's really only one particular reason why I'm talking to y'all about this. There's a kind of new fad like within the exercise like adaptation world. Really, like Quinn got me thinking about it, talking about like Hector Lake and saunas. Does anybody like saunas? Right? Have you ever like read anything about how saunas are like beneficial and good and stuff? Right? Kind of like a lot of people are like getting into saunas to like promote recovery from this, that, or the other thing. Right? Yeah? No? Okay, so how does that work? So we have particular like stress proteins that are actually made whenever we have like a given heat stress, or there's cold stresses too where this happens. But what I want you to know about is something called heat shock proteins. Heat shock proteins. So this is one of the earliest studies that I could find related to heat shock proteins. It was, I believe it was done in Iowa. Yeah, that's what it says. Yep, done in Iowa. So here's how heat shock proteins work essentially. So hopefully everyone recalls like translation, we make new proteins like at the ribosome. Whenever we start getting heated up, right, like then proteins denature. Is everyone good with this, right? So has anyone ever fried an egg, right? Like whenever you fry an egg, how the like egg white starts changing, that's because the albumin or the protein in there is getting denatured and getting changed. Now, if you get really hot, that can happen to you, right? So like it can, like people die from the heat all the time, but we actually make these really cool things called heat shock proteins to where we have a protein that is like folded in a particular way, we have a heat stress, it gets unfolded, so meaning it's non-functional. It's been denatured is what that's called. Then a heat shock protein will go and fold that protein back so that it works just like how it did prior to getting denatured or cooked. Is everyone good with this? So the idea is if you not train, but if you go into a sauna frequently, you'll have more heat shock proteins. Is everyone good with this?
Why do you want more?
So that like whenever you exercise in like a hot environment or like whenever you start like contracting muscles a lot, that increases muscle temperature. And it overall just makes all of the different parts in your cell more robust. So it can take more damage effectively. What's up, Quinn?
How long does it take to get adaptation? How long would you have to go to the sauna?
What was this study? Intervention, heat stress. On separate days, we obtained. Gosh. So if you remember, like the mRNA, like stimulations, you can get mRNA or like messenger, RNA of these heat shock proteins up quite a bit, like just within a session or two. But I, I'm kind of guessing here. But oh yeah, well, if you don't continually have the heat stress, then the change won't be permanent. But probably around 4 weeks would be where you would really see some positive results. And so right there, that's an educated guess. Don't take my word on that. Just generally, most physiological systems take approximately that long to adapt and get really well. I'll have a better answer for you next time. Cool. So, yeah. All right, cool. There we go. Heat shock proteins, pretty cool stuff. Like these other sets of slides, we've already talked about the cell signaling cascade for protein synthesis. So we're not going to talk about it again. So read through these if you want to. There's a couple of cool things going on in here. But how I talked about in like exercise genetics, I prefer that way, how it's spoken about a little bit more. But this is in your book. I think it's fairly interesting. I've got about 10 minutes. So let's go ahead and start getting into bioenergetics just a little bit. So those of you that like haven't printed out the slides for this or you don't have it on your computer, that's okay. Like I'm probably only going to cover like a couple of slides. And really any of these slides should be relatively easy for most of us to get through. So like we're going to talk a little bit about like the cell membrane, nucleus, mitochondria, a bit about like coupled reactions, endergonic, or anabolic or exergonic or catabolic reactions. So really just a lot of like new terminology that you may have not used prior to this. And then like how like ATP keeps on going. So first thing, metabolism. Whenever we talk about this, it's essentially just the summation of all the chemical reactions that occur in the body at any given moment in time. And there's overall 2 different flavors of reaction. The first one being anabolic, and ana is from the Greek meaning to build, so building or synthesis. That's what I want you to think about whenever we talk about anabolic or anabolism, building. Next thing, catabolic. Catabolic reactions, this is the breakdown of molecules. So overall, whenever we're talking about this in regard to exercise, anabolic reactions are going to be done in recovery and adaptation. That's mostly what's going to be happening. So like building new muscles, new mitochondrial proteins, new mitochondria even. Catabolic reactions are mostly going to be taken care of whenever we begin exercising. The breakdown of glucose, the breakdown of fatty acids, all that type of stuff. So next thing, bioenergetics. This is just overall the converting of food, right, into energy. So there's a level of like chemical energy, like within the chemical bonds. So like the hydrocarbon chains, for example, in fats, that we have to loose them from their chemical energy to actually get mechanical work, meaning muscle contractions. And like we can also get a fair bit of like chemical energy from ethanol or alcohol, which I believe like in or around St. Patrick's Day, I'm going to have you read a paper about that. There's actually some flavors of exercise that alcohol are pretty good for, and I'm going to tell you about it. So, all right, cell structure. Here, we've already talked about some of this, but just to like remind you, like some of the words I'm going to be using. Cell membrane or sarcolemma. That. Semi-permeable, what that means effectively is things that are lipids or fats or cholesterol, they more or less readily get into the cell without a transporter. Now things that are more like soluble in water, they typically need a transporter. Unless it's water and it's osmosis, or it could use aquaporin or something like that. But that's really just what I want you to know about that so far. Nucleus, we've talked about that so much it's ridiculous. Cytoplasm, also called cytosol, sarcoplasm. It sounds a lot like, what is it, Ghostbusters type of stuff? Right? No? Anybody? Doesn't it? It doesn't, Brad? That's okay. It does to me. All right. So here we go, just a little bit like another picture like taken from your book about how all of this works together. And just really trying to work on conceptualizing how all of this is. So a muscle cell, which is probably what we're the most concerned with, if you don't know, they're multinucleated, meaning that there's more than one nucleus within them. And that's going to be really important whenever we start talking about resistance training adaptation, and like the myonuclear domain theory. But mitochondria, we just have them floating around like different beans. And actually, it's at a conference one time, I saw pretty convincingly that it's actually like a reticulum that just permeates the entire cell. So like how we think of it as like a bean, like it might not be that, but I don't know. I've actually isolated mitochondria from like rat muscles, and like they took up oxygen, so like after they died, so that's pretty cool. But that's essentially how that looks. Okay, next thing to just generally understand. All energy on Earth comes from the sun. So like photosynthesis like happens, effectively plants do almost the like reverse of cellular respiration, what we do. And they take in carbon dioxide, they make glucose, and then animals or us eat them, and then we get more energy out of it. And overall, the chemical energy begins in the sun. So I think that's pretty cool. Next thing here, bioenergetics, overall, it's just the process of converting chemical energy to mechanical energy. And this picture right here, I think, is pretty cool. So we'll talk about like efficiency and how much we actually lose to heat, like endotherms and things. So carbohydrates, fats, others, we make ATP, we have various metabolic functions. Then some waste comes off, carbon dioxide or water, metabolic water, that's actually at the very end of the electron transport chain. And then we lose quite a bit of energy as heat, like making sure that we don't freeze to death outside. Here, let's see. How many more? Oh, gosh. Yeah, here, let's have this slide be our last one today. All right. So cellular chemical reactions. How I said there's anabolic and catabolic, here's new terms for those. Endergonic or endothermic. We can think about these as anabolic reactions as well. Exergonic reactions, these release energy, exothermic or catabolic. So essentially how I want you to think about this is the complexity of different molecules. So in, say, an endergonic reaction, whenever like we eat protein, we have to break it down to amino acids so it can circulate in our blood. Those amino acids get into our muscles, and then we start putting them together in interesting ways to make new proteins or grow muscle, say. So that's going from like a less simple thing, a more simple compound to a more complicated compound. So like leucine is fairly simple, bicep protein is fairly complex. Hopefully everyone understands that. Now, that's essentially what I want you to think about with endergonic reactions. Now, exergonic reactions, these are the release of energy. So this, like muscle glycogen, hopefully everyone knows that like humans have this, right? We take glycogen, which is a fairly complex molecule, and we break it down into smaller and smaller things through glycolysis and the citric acid cycle and things in order to release energy from it. And that, we'll probably start back on this slide next class. Any questions about anything? Good luck, have a good weekend. And I don't know, I think it's going to snow tonight.